Hello dear students, this is a video on practical hematology and it deals with the dilution of blood samples. This is a very narrow scope that I have chosen for this particular video. So we will see the dilution of blood sample, diluting pipettes, dilution factor and diluting fluid. Before we actually start, let me make a point. These methods are almost obsolete in the real world. I mean, in the real pathology laboratories, these methods are hardly in use anymore. However, in many medical schools, particularly in India, we still see these methods being taught at first year medical level, and therefore I thought I'll take up this topic and see what errors are made by the students. Right, so having said that, now let's talk about the dilution of blood. These pipettes are used to dilute the blood sample in case of the RBC count or WBC count, or even for platelet count or reticulocyte count, but we are going to focus on RBC and WBC pipettes. The question is, why do you need the dilution of blood? I mean, why do you have to dilute the blood sample for the RBC or WBC count? Consider this. The RBC count is generally about 5 to 6 million cells per cubic millimeter of blood. 5 to 6 million cells, 50 to 60 lakhs of cells in 1 cubic millimeter of the sample or 1 cubic millimeter space. That's too much. That will be too much crowding of cells, and therefore it will be humanly impossible to count those many cells in that small a space. Virtually impossible. So what we actually are up to is that we will first dilute the blood sample, means, now note this word, we are going to disperse the cells. Dilution of blood sample means dispersion of cells so that we can count them manually under the microscope. Alright, that's the reason why we need dilution of the blood sample. Now remember, we are dispersing the cells by diluting the blood. If the dilution factor is 200, that means we diluted the blood sample 200 times, and therefore eventually once we count the cells and all other calculations are done, then we will multiply by 200. The eventual count will be after multiplication by 200. Why? Because by diluting the blood 200 times, we were actually getting those cells 200 times less, isn't it? The cells were dispersed in the sample. How much dispersion? By 200 times. And therefore, eventually, to get the actual count, we multiply by 200 to get the actual cell count. So that's the reason that we dilute the blood samples. Now coming to the pipettes used for the dilution of blood sample. These two pipettes can be seen here in the picture, the RBC pipette and the WBC pipette. Now, why do we need two different pipettes for RBC and WBC count? Can we not use just one dilution pipette? No. The reason is WBC count is 4 to 11,000 per cubic millimeter of blood. RBC count is 5 to 6 million cells per cubic millimeter of blood. See the number, see the difference. And therefore, in the case of WBC count, a lesser dilution of blood would be acceptable. 4 to 11,000 per cubic millimeter of blood. So we can just dilute the blood 20 times and we will be able to count the cells, WBCs. But in the case of RBC count, RBCs, they are in millions, 50 to 60 lakhs or 5 to 6 million per cubic millimeter of blood. So we will need a greater dilution to disperse those cells so that we can count them under the microscope, manually count them under the microscope. And therefore we need 200 times dilution. So remember, dispersion will have to be greater in the case of RBC count, because RBC number is much larger, much greater as compared to WBC count. However, we use these pipettes for 20 times dilution or 200 times dilution in the case of WBC count or RBC count respectively. All right. So that was another point to be highlighted. Why do we need lesser dilution for WBC count and higher dilution, greater dilution for RBC count? That's only because of their respective numbers. Right. Now, these are the pipettes which are used. Well, let me stress on one more point. Although we are calling them as RBC pipette and WBC pipette, and RBC pipette is going to cause dilution by 200 times, WBC pipette causes dilution of blood by 20 times, but these are simply dilution pipettes. I mean diluting pipettes. They can be used to dilute any fluid by either 20 times or 200 times. Okay, we are using it for blood sample dilution, but they can be used for other purposes. Semen sample dilution, for instance, these pipettes can be used. Anything, any fluid, if you have to dilute it by 20 times or 200 times, we can very well use these pipettes. Right. Now, coming to identification of the pipettes. How do you identify RBC pipette and WBC pipette? Look, if you look at the pipette, there is a bulb provided in the pipette. In the RBC pipette, the bulb is larger. The WBC pipette, the bulb is relatively smaller. And the reason is obvious, because in RBC pipette you need a greater dilution. WBC pipette you need a lesser dilution. Second point, there is a bead inside those pipettes, inside the bulb actually. Red bead inside the RBC pipette, RBC bulb, white bead in the WBC pipette or WBC bulb. So that's quite obvious. Red for the red blood cell count, white for the white blood cell count. Simple. Third point is one end from where we just take the blood sample or suck the blood sample. That end is red for the RBC pipette, white for the WBC pipette. But that color coding is generally lost over a period of time. In many labs, you will see those ends or mouthpieces have been changed frequently, so that color coding is lost. So you can't depend on that. Anyways, last but the most important one is the markings on the pipettes. Although not visible clearly in this particular picture, markings on the WBC pipette are 0.5, 1 and 11. On the RBC pipette, the markings are 0.5, 1 and 101, 101. Now, note this point here and then I'll explain it again later. Below the bulb, it's 1 and above the bulb is 11. That means the capacity of the bulb is 10. Obvious. 1, 11. So the capacity of the bulb itself is 10 in the case of WBC pipette. And for the RBC pipette, below the bulb 1 and above the bulb 101. So capacity of the bulb is, come on, capacity of the bulb will be 100. Yes, that's right. 1 below the bulb, 101 above the bulb. That means this is 100 parts, right? So that's the capacity of the bulb. Now, let's see how the procedure is actually carried out. One more point here. Are there any units to these numbers? 0.5, 1 and 11 or 0.5, 1 and 101. Are there any units, microliter, ml or whatever? No. Um, there are no units to these numbers. I repeat, there are no units to these numbers. This is simply dilution. I mean, one part of blood will be diluted up to 200 times. One part of blood will be diluted up to 20 times in the case of WBC. So, there are no numbers actually. These numbers don't have any units, I mean to say. I beg your pardon. No units to these numbers. Any sample, any fluid can be diluted up to that extent. Okay. Now let's see the procedure and try to understand what goes wrong with the procedure and how the students answer some questions incorrectly. Let's say RBC pipette. We will take the blood sample up to 0.5 mark, up to the 0.5 mark, and then we start taking the diluting fluid. Mark this procedure carefully. We take the blood up to 0.5 mark in the pipette, and then immediately we start taking the diluting fluid. What happens is, the blood sample which was there, it goes into the bulb, and then the diluting fluid also starts going into the bulb. So fill it, fill it, fill it, fill it till that mixture has come up to 101. That means you have filled the entire bulb. What does the bulb contain? The blood sample and the diluting fluid. So it's calculated like this. 0.5 was the 0.5 parts of blood were taken and diluted inside the bulb. What was the capacity of the bulb in the case of RBC pipette? Come on, capacity of the bulb? 100, yes. So when you have a mixture inside the bulb where blood is getting mixed and diluted, it means 0.5 parts of blood were diluted to 100. Capacity of the bulb is 100. So 0.5 parts of the blood diluted to 100. That means 1 part diluted to, come on, complete the sentence. 1 diluted to 200. And then remember, when you are going to use that mixture, don't forget to discard first 1 or 2 drops when you are going to use the mixture. Because you know what has happened? Blood sample has gone into the bulb entirely, and the diluting fluid has also gone there, and mixing happens over there. That means this portion, the stem portion, will contain only the diluting fluid, no blood, no cells. And therefore, when you are going to use it, first discard 2 or 3 drops. Discard this portion, I mean this fluid in that portion should be discarded, and then the mixture will start coming out. So you can charge the Newbar's chamber with that mixture. Remember this. I repeat once again, because very often even the tutors tend to go wrong while explaining this. You know what is the error made by some tutors and therefore the students is that they tell the students 0.5 was diluted to 101, therefore 1 part is diluted to 202, and then discard the first 2 drops to make the dilution factor as 200. No, that's incorrect explanation. 0.5 was diluted to 100 because dilution is taking place within the bulb, and capacity of the bulb was 100, out of which 0.5 parts were blood. Okay, therefore 0.5 was diluted to 100. That means 1 is diluted to 200 times dilution. And because entire blood sample was mixed in the bulb and this portion will contain only the diluting fluid, therefore you discard these first 2 drops from the stem. Many tutors teach the other way around, that 0.5 diluted to 101, so 1 diluted to 202, and therefore you discard the 2 drops to make it 200. No, that's wrong. Okay, in the case of WBC pipette. Take blood sample up to 0.5 mark. Now this is easy to understand. Take the blood up to 0.5 mark, then start taking the diluting fluid. Blood sample goes in the bulb, diluting fluid starts going into the bulb. Fill it, fill it, fill it, fill it, fill it, and eventually it will get filled and it will reach up to 11 mark in this case. So that means now the blood sample and the diluting fluid is entirely within this bulb. What was the capacity of the bulb in the case of WBC diluting fluid, WBC pipette, I beg your pardon. WBC pipette, the bulb had a capacity of 10. That means 0.5 is diluted to 10 parts. That means 1 diluted to 20. Yes, therefore, blood will be diluted by 20 times, and that's in the case of WBC pipette. And as I said, first 2 drops will have to be discarded before you charge the Neubauer's chamber, because this portion will contain only the diluting fluid. All right. One more point. What is the function of the bead inside the bulb? One function or one thing about them is that identification mark. Red bead for red blood cell count and white bead inside the WBC pipette or WBC count. Apart from that, the bead is actually given for mixing purpose. Remember this. The glass walls are smooth from within, and therefore mixing of the blood sample with the diluting fluid would not have been proper. So this bead actually helps in the mixing of blood sample with the diluting fluid, and the dilution, I repeat again, nth number of time, that the dilution takes place only and only within this bulb. All right. So that's about the diluting pipette and how much dilution. Sometimes the teachers also ask this question: can you use RBC pipette for WBC count? Now remember, by now you must have understood the concept. WBC count is in thousands, therefore we dilute the blood only up to 20 times. RBC count is in millions, therefore we dilute the blood up to 200 times in the case of RBC count, because millions of cells in 1 cubic millimeter, therefore greater dispersion needed. If you have understood this, then the answer would be easy for this question. Can you use RBC pipette for WBC count? Yes, we need to use RBC pipette in the case of WBC count in the conditions of leukemia. You know, in the condition of leukemia, the WBC count is very, very high. It can go almost in millions. And if that is the case, maybe not in millions, but a few lakhs, 500,000 or something like that. And if that is the case, the WBCs will be crowded too much in 1 cubic millimeter of blood. Therefore, you will need a greater dispersion. And in that case, you will use RBC pipette to count the WBCs. So remember, whenever the WBC count is too high, example I gave you, leukemia, we will have to use RBC pipette to count those WBCs. So basic concept is simple. More the cell number that means greater crowding of the cells, you will need a greater dispersion and therefore the greater dilution factor. That's the dilution of blood sample. Now finally, what are the contents of the diluting fluid? Here I have seen many students give some wrong answers during the Viva. Therefore, I take up that part as the last part of this particular video. Contents of the diluting fluid. First of all, RBC diluting fluid is called as Heinz fluid. Heinz fluid. And the WBC diluting fluid is called as Turks fluid. So, Heinz fluid, Turks fluid. Let's see the contents of the diluting fluid. In the case of RBC diluting fluid, it's called as Heinz fluid, as I mentioned almost thrice now. And the contents: NaCl, sodium chloride, 0.9%. You know, we are talking about the RBCs, and therefore the fluid that surrounds the RBCs, our diluting fluid, it has to be isotonic. Isotonic to what? Isotonic to the fluid inside the RBCs. And therefore 0.9% sodium chloride is the isotonic fluid. It provides isotonicity to this entire fluid. You know, if it is not isotonic, if the fluid surrounding the RBCs is not isotonic, then there will be fluid movement either into the RBCs or out of the RBCs, and then RBCs will be broken down. We won't be able to count them. So NaCl, next sodium sulfate. Sodium sulfate. Now here many authors have suggested that it acts like an anticoagulant. Let me make a point. When blood is diluted 20 times or more, listen to this part carefully. When blood is diluted 20 times or more, you don't need an anticoagulant in that blood sample. If the blood is diluted 20 times or more, you don't need to add anticoagulant in that sample. Why is that? Well, because if the blood dilution is too much, if the dilution is too much for the blood sample, that means the clotting factors present in that sample will not be able to come together and form a clot. They are dispersed, just like the cells get dispersed. Even the clotting factors will get dispersed too much. They will get diluted too much, and therefore clot formation will not occur. And therefore you don't need really the anticoagulant. RBC diluting fluid, we are diluting the sample 200 times. So clot will not happen, clot will not occur. Then what is why is this sodium sulfate added? It is anti-rouleau formation. Sodium sulfate will act as a anti-rouleau formation. You know, anti-rouleau formation. You know, when the blood is taken outside the body and made to stand still in a vertical tube, RBCs, they come together to form clumps which are called as rouleaux. So here we are taking the blood sample out of the body. RBCs may form the clumps, they will form the rouleaux, and we need to prevent that because we are going to count the RBCs, and therefore we need an anti-rouleau forming agent, that's the sodium sulfate. Third is an interesting one, mercury chloride. Mercury chloride. Many authors have simply written in front of this as preservative. Well, yes, it is a preservative, but it preserves what? I ask this question very often in my exams, in the vivas. Preservative, mercury chloride, preserves what? Students are clueless. Okay, does it preserve that slide that you have made, the Dubas chamber or that sample? Well, we don't need a preservative for that. We are going to use it only for 5-10 minutes and going to discard it after that. So we don't need a preservative. Then preservative of what? Preservative of the entire solution. Look, we make the solution for the entire batch of 50 students. we may make it 15 days before the actual practical, and we will keep it in some glass container. So since it contains sodium chloride and the other constituents, there may be bacterial growth or microbial growth in that diluting fluid. We have made it in bulk, and we are keeping it in the lab. So it will act as a preservative. It will prevent the growth of bacteria and other microbes if we are going to keep it for 15-20 days. So remember, preservative of the entire fluid. Let's say you have made 20 liters of the diluting fluid before the practicals, and you are going to keep it in the lab for next 10-15 days. That will be preserved. So remember this, it preserves the diluting fluid itself. Okay, and many students tend to answer preservative of RBCs, preservative of the Newbar's chamber. No, preservative of the fluid. And then water to add up to the volume. That's the Heinz fluid and its contents and the functions of those contents respectively. Let's see the WBC diluting fluid. It's called Turks fluid. It dilutes the blood. We are going to use it to dilute the blood 20 times. What are the contents and the functions of those contents? 1% glacial acetic acid. What is the function of this? Look, glacial acetic acid is going to break the membranes. It's going to rupture the membranes of the cells. Are you with me so far? We are now talking about the WBC diluting fluid. Okay. WBC diluting fluid will be used to count the WBCs. And you know, there are going to be lots of RBCs in that sample. We don't want those RBCs to interfere with our count. And therefore, glacial acetic acid 1%, it will break open the membranes of RBCs and to some extent also the WBCs. But mainly this is to lyse the red cells, to lyse the RBCs, to break down the RBCs, so that RBCs do not create an interference in our counting of WBCs. We are counting WBCs, so we break down and remove the RBCs. That's glacial acetic acid. But as I said, it can also break membranes of the WBCs as well. And then we have a stain, methylene blue, because what we will mainly see under the microscope will be the nuclei of WBCs. You know, WBCs are nucleated cells, unlike the RBCs which are non-nucleated. So we will stain those nuclei with methylene blue, and then we will start counting the WBCs, and then water to add up for the volume. These are the contents of the WBC diluting fluid, the Turks fluid. So this, in short, is about the dilution of blood sample, diluting fluid, its contents, why do we need the dilution, and so on and so forth. So in the next video, we will discuss about some other practical.